Chapter 7, Section 12, Regulatory RNAs. Here we're going to look at RNAs, and up till now, whenever somebody says RNAs, okay, first thing you think about, messenger RNA. Think a little harder, ribosomal, transfer RNAs. These are what are referred to as non-coding, the ribosomal RNA, the tRNA. Okay, unlike messenger RNA, which is the blueprint on how to, basically put together a polypeptide, our RNAs and tRNAs don't carry information. They do work. They're there for other things. So these non-coding RNAs, okay, ribosomal, transfer, and there's also what they call signal recognition particle RNAs. These are going to be short RNA strands that will bind to and interact with, receptors, signal proteins, other nucleotide strands to, increase, decrease, and, activity and things like that. We also see that there's what are referred to as small RNAs. You'll see this in the literature as sRNA. Small RNAs are anywhere from 40 to 400 nucleotides, very small. These will regulate gene expression. We find these in both prokaryotes and eukaryotes. What they do is they bind directly to messenger RNAs. How's that good? How's that bad? Well, think about it. In your body right now and in a cell, bacterial cell, there are no double-stranded RNAs. We have nucleases, proteins that hunt down. They enzymatically cleave and chop up any double-stranded RNA because over 1 billion years of evolution, our cells, prokaryote, eukaryotic, have come to find, recognize double-stranded RNA as being viral in origin. So bind that up, bind these short things, small RNAs to a messenger RNA, and you can turn over the messenger RNAs much, much faster. Or you can bind and you can regulate. Small fragment up near the 5' end, may interfere with the binding of the small subunit. May interfere with binding of the large subunit. Translation inhibited. Or it may inhibit that complementary self-binding, that self-complementary binding we talked about in section 11 with the heat shock proteins. Well, that may increase or decrease stability of an RNA strand. So, as I just mentioned, when it comes to these small RNAs, they have different abilities, different means of altering, translation, the making of the polypeptide. As I said, they can block by binding, interfere with up at the, binding of the ribosomes up near codon 1. Okay, can't get to codon 1, can't get the, that special F-met initiation tRNA to bind. Well, then guess what? No translation. Can also increase expression by complementary base pairing, changing the stem loop pattern, which now means the ribosome binding site is now open for the small subunit and the large subunit to come, bind, and start translation. Either block or help. Increase, decrease degradation based on just how much of this small RNAs are binding, where they're binding. Those nucleases will either ignore it or, mistakenly recognize it as double-stranded viral RNA. So here's just the figure illustrating what I just talked about. I'll let you look at those in the textbook. We see that there are numerous types of these small RNAs. They have different jobs. They're going to be expressed under different conditions. Under different conditions, due to oxidation, iron presence, glucose phosphate stresses, quorum sensing, all sorts of different things will cause increase, decrease in these sRNAs, or I shouldn't say in total, in different ones, because there's more than more than one going on in your cells or in bacterial cells at any given time. So changes in iron, changes in glucose, changes in oxidative stress, quorum signals will change the different sRNA levels at any given moment. Here's an example from the textbook showing that, the SGRS and this PTSG, this signal, and this one here. We also have other regulatory small RNAs, regulatory ones. These are known as trans sRNAs. These trans sRNAs, they're going to be in the chromosome, and they're going to be in the DNA sequence between genes, okay? Unlike us, eukaryotes, who we have, introns, exons, and there's lots of junk in between our genes. Well, we find that with prokaryotic chromosomes, genes, especially if they're in operons, are going to be butted up together. Well, some of the times there's going to be a little bit of a space in between the operons, in between individual genes that are not operon-based. That's where you're going to find these trans sRNA sequences. What they can do is they can bind to proteins. They can bind to other nucleic acid strands and increase, decrease expression, increase, decrease stability. We find that the trans sRNAs use the protein at HFFQ. HFQ acts as a chaperone. It will bind to the small regulatory, the trans here. The trans will then go and bind a specific nucleic acid sequence. Here's a regulatory sequence on a messenger RNA. Yes, even messenger RNAs are going to have regulatory sequences where there's either going to be some form of a stem loop for self-complementary binding, which will increase or decrease ribosomal interactions, or you can have these bind, keeps the messenger RNA from binding with itself, and it also can form stem loops that will recruit in different, proteins and whatnot that are going to increase, decrease translation of this gene on this messenger RNA. I know it's very vague, but unless you know specifically what, which gene is on this messenger RNA or which operon, and without knowing specifically which of these trans sRNA sequences we're discussing, I have to be pretty vague.